Hello students, welcome to EPG Pathsala. I am Samar Singh from Department of Microbial Biotechnology, Punjab University. Today we are going to discuss about a module named DNA polymerases under paper Genetic Engineering and Recombinant DNA Technology. Dear students, after going through the module you would be able to tell what are the DNA polymerases, their discovery, the types of DNA polymerases found in E. coli, what they do, what's their structure, conservation and its function, DNA polymerization reaction and its attributes, DNA polymerases fidelity and processivity. Also, you are going to learn how many DNA polymerase families are there and the examples of each of them, how special function DNA polymerase telomerase carries out lengthening of a linear DNA ends. DNA polymerases are the enzymes that catalyze the DNA polymerization reaction in a cell. They generally catalyze the reaction which involves the transfer of a nucleotide monophosphate from a nucleotide triphosphate to a pre-existing polynucleotide primer molecule, which can be either a DNA or a RNA molecule, finally resulting in the extension of a polynucleotide chain as depicted in the shown reaction summary. The example of DNA polymerases which use DNA as a template, or in other words the DNA polymerase activity in a DNA template dependent manner, are DNA pol 1, DNA pol 2, DNA pol 3, PFU, TAC, etc. The examples of DNA polymerases that use RNA as a template, or in other words the DNA polymerase activity is RNA template dependent, also referred as reverse transcriptases, are telomerases and reverse transcriptases from retroviruses such as MMLV or Molony-Murine leukemia virus, etc. There are also DNA polymerases that show template independent DNA polymerase activity such as terminal deoxynucleotide transferase enzyme. DNA polymerases are the enzymes that catalyze the DNA polymerization reaction in a cell. They generally catalyze the reaction which involves the transfer of a nucleotide monophosphate from a nucleotide triphosphate to a pre-existing polynucleotide primer molecule, which can be either a DNA or a RNA molecule, finally resulting in the extension of a polynucleotide chain as depicted in the shown reaction summary. The example of DNA polymerases which use DNA as a template, or in other words the DNA polymerase activity in a DNA template dependent manner, are DNA pol 1, DNA pol 2, DNA pol 3, PFU, TAC, etc. The examples of DNA polymerases that use RNA as a template, or in other words the DNA polymerase activity is RNA template dependent, also referred as reverse transcriptases, are telomerases and reverse transcriptases from retroviruses such as MMLV or Molony-Murine leukemia virus, etc. There are also DNA polymerases that show template independent DNA polymerase activity such as terminal deoxynucleotide transferase enzyme. DNA polymerases are enzymes that make copy of DNA. They are mainly responsible for accurately and efficiently replicating the genome to ensure the maintenance of genetic information and bring about its faithful transmission through generations. First evidence of the existence of an enzymatic activity that is capable of synthesizing DNA came from the work of Arthur Kornberg and colleagues, who first purified and characterized the first DNA polymerase, which was a single polypeptide enzyme, from E. coli cells, which later also came to be known as DNA polymerase 1 or Pol 1. Later, investigators found that E. coli contains at least 4 more other distinct DNA polymerases. Later studies of DNA polymerase 1 revealed features of DNA synthetic process that are now known to be common to all DNA polymerases. DNA polymerases using one DNA, or in other cases they may also use RNA in case of reverse transcriptase, strand as a template, make a complementary copy, thus allowing stable passage of the information from one generation to the next. There are multiple DNA polymerases. For example, more than 90% of the DNA polymerase activity observed in E. coli extracts can be accounted for by DNA polymerase 1, but later on in 1955, however, evidence began to accumulate that it is not suited for the replication of the large E. coli chromosome. The evidences in support of the existence of more DNA polymerases are: first, the rate at which it adds nucleotides, which is usually 600 nucleotides per minute, is too slow by a factor of around 100 or more to account for the rates at which the replication fork moves in the bacterial cell. Second, DNA polymerase 1 has relatively low processivity. Third, genetic studies have demonstrated that many genes and therefore many proteins are involved in replication, like DNA polymerase 1 clearly does not act alone in this process. Fourth, and most important, in 1969, John Cairns isolated a bacterial strain with an altered gene for DNA polymerase 1 that produced an inactive enzyme. However, this strain was abnormally sensitive to agents that damaged DNA. It was nevertheless viable. A search for other DNA polymerases led to the discovery of E. coli DNA polymerase 2 and DNA polymerase 3 in the early 1970s. DNA polymerase 2 is an enzyme involved in one type of DNA repair. DNA polymerase 3 is the principal replication enzyme in E. coli. This table shows the comparison of three DNA polymerases of E. coli. As you see, DNA polymerase 1 has 3' to 5' exonuclease or proofreading activity, as well as 5' to 3' exonuclease activity. While if we look at DNA polymerase 2, it just has 3' to 5' exonuclease activity, but no 5' to 3' exonuclease activity. But its processivity is almost 7 to 8 times that of DNA polymerase 1. And if we look at DNA polymerase 3 enzyme, it just has 3' to 5' exonuclease activity, no 5' to 3' exonuclease activity, but its processivity is much higher. It's more than about 5 lakh bases. Now let's talk about structure and function of DNA polymerase. The structure of DNA polymerase core that contains the catalytic site for nucleotide addition generally folds into three structural domains that roughly resembles a right hand. The uncanny similarity in appearance has led to the three domains being called the palm, the fingers, and the thumb. The DNA polymerase core has been found to be the most highly conserved component of the polymerase enzyme complex in both multiple as well as single subunit enzymes. Structurally, the palm domain is the most conserved domain that forms a cleft into which the growing double-stranded DNA fits, and it has the polymerase active site too. The finger domain of the DNA polymerase help warp the single-stranded template strand and positions the incoming DNTPs in relation to the template DNA. The thumb domain helps to hold the elongating duplex DNA and maintains the contact with the template, allowing processive synthesis of a DNA molecule. The 3' to 5' exonuclease function of DNA polymerase that removes incorrect bases incorporated whenever present is found as an additional domain near polymerase active site on the palm domain. DNA polymerase reaction. Its fundamental reaction is a phosphoryl group transfer. Here, the 3' hydroxyl group of the nucleotide at the 3' end of the growing strand acts as a nucleophile, attacking at the phosphorus of the incoming deoxynucleoside 5'
triphosphate. For that, you can look at the next slide. Inorganic pyrophosphate is released in this reaction. The general polymerization reaction can be summarized as DNMPN plus DNTP, that makes DNMPN plus 1 plus 1 pyrophosphate is released. Here DNMP and DNTP are deoxynucleoside 5' monophosphate and 5' triphosphate. Now let's talk about polymerization and elongation of a DNA chain. As you see, DNA polymerase requires a single unpaired strand to act as a template and a primer strand to provide a free prime hydroxyl group at 3' end, to which a new nucleotide unit is added. Each incoming nucleotide is selected in part by base pairing to the complementary nucleotide in the template strand. The new free 3' hydroxyl available on the reaction product usually allows the addition of another nucleotide. Here the two magnesium ions that you see in the cartoon, which are coordinated to the phosphate groups of the incoming nucleotide triphosphate and to 3 aspartic residues, 2 of which are highly conserved in all DNA polymerases, plays essential role in the polymerization reaction. The magnesium ion towards exterior on the right side facilitates the attack of 3' hydroxyl group of the primer on the alpha phosphate of the incoming nucleotide triphosphate. The other magnesium ion facilitates the displacement of the pyrophosphate. Both magnesium ions supposedly stabilize the structure of the pentacovalent transition state. DNA polymerization reaction facts. Remember, DNA polymerase makes a complementary copy of one DNA strand. Its template dependence and Watson-Crick base pairing rules are always followed. All DNA polymerases require a template DNA or RNA. The polymerization reaction is guided by a template strand according to the base pairing rules predicted by Watson and Crick. Primer requirement. All DNA polymerases require a primer, whether it is RNA or a DNA. And a primer is a strand segment complementary to the template with a free available 3' hydroxyl group to which a nucleotide can be added. The free 3' end of the primer is called the primer terminus. In other words, part of the new strand must already be in place before the DNA polymerase can act, and all DNA polymerases can only add nucleotides to the pre-existing strand. Specialized enzymes synthesize primers whenever they are required. DNA polymerization reaction facts. After adding a nucleotide to a growing DNA strand, a DNA polymerase either dissociates or moves along the template and adds another nucleotide. Dissociation and reassociation of polymerase limit the overall polymerization rate. That is, the process is generally faster when polymerase adds more nucleotides without dissociating from the template. The average number of nucleotides added before a polymerase dissociates defines its processivity. DNA polymerases vary greatly in processivity, such as a few to many thousand nucleotides. DNA polymerases fidelity and processivity. The processivity of replicative polymerases is very important for the genome replication. It is usually achieved by extra domains or subunits that modulate the dissociation of DNA polymerase from DNA. For this, kindly refer to comparison of DNA polymerases of E. coli. The accuracy of DNA polymerization to make a complementary copy of the template DNA is known as fidelity. It is of utmost importance for replicative DNA polymerases and to the replication of genome. Errors in DNA synthesis needs to be minimized because changes in the genetic code will be passed on to the progeny cells and would alter the essential cellular functions. A replicative polymerase typically makes one uncorrected error for every 10 to 5 to 10 to power 6 nucleotides added. DNA polymerases fidelity. This remarkable fidelity of replicative DNA polymerases is achieved by activities ensuring the identity of nucleotides at two distinct stages during polymerization and post-polymerization. During the polymerization reaction, selection of correct nucleotides, the spatial geometry of the active site allows selective accommodation of correct complementary nucleotides as they fit precisely into the active site when base paired with the template strand. That is, the pairing of A with T or C with G will be preferred over mismatches as the geometry of an adenine to thymine pair and the cytosine to guanine pair are similar to each other, allowing it to fit neatly in the active site, whereas mismatched nucleotides will have a different geometry that does not fit as well. The correct geometry favors the catalytic reaction and the addition of the incoming nucleotides onto the new DNA strand. This cartoon shows how this process functions. As you see in the right hand side, here the pink color shows the incorrect pairing of bases, while those shown in the gray show the correct pairing of the bases. So based upon whether the incoming base is correctly matched, the polymerase reaction moves on, or it allows the DNA polymerase to correct it before it moves on.
DNA polymerases also achieve further level of fidelity after polymerization reaction. That's also usually referred as proofreading. What happens in this? The selective removal of the last misincorporated base to the growing polynucleotide chain at polymerase active site is achieved by specially separated 3' to 5' exonuclease active site. What happens whenever a correct base are being added, the polymerase keeps adding new nucleotides and relocates to next template position. But if a mismatched base gets incorporated or misincorporated, the polymerase activity is slowed down, allowing the growing polynucleotide chain to be relocated to the exonuclease site, which then removes the terminal nucleotide. Then the chain is again shifted back to the polymerase active site to resume polymerization of the polymerase at the polymerase site. For this, please refer to the structure of polymerase that we have discussed earlier.
This cartoon displays how the DNA polymerase fidelity or proofreading or editing works. Here, as you see in the cartoon, the incorrect base pair incorporation at the growing strand, which is shown in pink, at the 3' end leads to mispairing at the 3' end, which is detected and the 3' end is moved to the 3' to 5' exonuclease site on the polymerase. This involves breaking of several base pairs between the nascent strand and the template to allow relocation of the 3' end to editing site, where the 3' end nucleotide is removed, followed by allowing the 3' end to return to the polymerase active site in the palm domain, where now the polymerase reaction can continue. There are multiple DNA polymerases. Cells have DNA polymerases to perform distinct specialized functions such as repair of DNA, continue replication across damaged DNA, replication of telomeres, etc. Beside the well-known replicative DNA polymerases, DNA polymerase 3 in bacteria or DNA polymerase delta and epsilon in eukaryotes that copy the bulk of DNA during replication. Polymerases may comprise just a single chain protein performing all range of functions such as nucleotide addition, proofreading and substrate specificity, or multiple subunits with each subunit having a distinct function. Based on amino acid sequence similarity and structure, DNA polymerases have been classified in 7 distinct families, namely A, B, C, D, X, Y, and reverse transcriptase. A family. Besides polymerase activity, they generally contain a 5' to 3' exonuclease domain that allows it to remove DNA or RNA ahead of it, and also features the 3' to 5' proofreading exonuclease activity. The well-known example is DNA polymerase 1 from E. coli. The family B, C, and D are replicative polymerases of eukaryotes, bacteria, and archaea respectively. These polymerases have high fidelity and possess a 3' to 5' proofreading exonuclease activity. X family of polymerases, they are specialized for DNA repair, filling in the gaps generated in the DNA during repair. Y family of polymerases, they are specialized to replicate past bulky adducts present in the damaged template DNA. Other polymerases will stop polymerization whenever they encounter such lesions, but Y family of polymerases are designed to not do so. Reverse transcriptases, they are specialized DNA polymerases that use single-stranded RNA as a template to synthesize a complementary DNA strand. The structure of reverse transcriptase is very much like that of other polymerases, and it has thumb, finger domains, and palm domain. The active site of reverse transcriptase is similar to that of other DNA polymerases and uses the same catalytic mechanism for linking nucleotides together, and they are mainly encoded by viruses as well as DNA elements called retrotransposons found in eukaryotic genomes. This is the comparative chart showing different DNA polymerase families and giving you example of the members and kind of function they perform in the cell. Telomerases. They are a specialized reverse transcriptase enzyme which are involved in replication of the telomere found in eukaryotic cells. Although the existence of this enzyme may not be surprising, the mechanism by which it acts is remarkable and unprecedented. It contains both the RNA and protein components. The RNA component is about 150 nucleotides long and contains about 1.5 times copies of the appropriate CA telomere repeat. This region of the RNA acts as a template for synthesis of the TG strand of the telomere. Telomerase thereby acts as a cellular reverse transcriptase that provides the active site for RNA-dependent DNA synthesis. Telomere synthesis requires the 3' end of a chromosome as a primer and proceeds in the usual 5' to 3' direction. Having synthesized one copy of the repeat, the enzyme repositions to resume extension of the telomere. The complementary CA strand is synthesized by cellular DNA polymerases, starting with an RNA primer.
Dear students, in this figure on your right hand side is a visual depiction of DNA synthesis by telomerases at the terminus of a linear DNA molecule in eukaryotes. The telomerase uses an internal RNA template that is shown in green background in the figure as a template to synthesize or add complementary sequences in 5' to 3' direction to the existing DNA end as indicated in step 1 of the figure. And having synthesized one copy of the repeat sequence, the enzyme then repositions, moves from one place to the next to resume extension of the telomere from a new position as indicated in step 2. The step 1 and 2 are repeated multiple times to keep lengthening the DNA strand in 5' to 3' direction. However, remember that the strand complementary to this telomerase lengthened strand is synthesized by the cellular DNA polymerases, starting with an RNA primer as in normal DNA replication. I would also like to remind students that telomerase is not a regular reverse transcriptase in the sense that unlike the retroviral reverse transcriptases, the telomerase copies only a small segment of RNA that it carries within itself and uses it as a template. Dear students, let's recap what we have learned so far in the current module about DNA polymerases. DNA polymerases catalyze the polymerization of DNA using a strand of DNA as template but the RNA and template independent DNA polymerization is not uncommon. The DNA polymerase structures are conserved, especially the polymerase domain, when present with other structural and functional domains and features. Based upon the sequence similarity and function, there are 7 families of DNA polymerase enzymes, namely A, B, C, D, X, Y, and reverse transcriptases or RT. The polymerase or polymerization activity of DNA polymerase is 5' to 3', but exonuclease activity whenever present could be either 5' to 3' or 3' to 5'. And the addition of bases is always done on a pre-existing polynucleotide tail with a free 3' hydroxyl group. The polymerases involved in replication of genomes, which are also known as replicative polymerases, generally have high processivity and high fidelity due to inherent selectivity for the correct complementary nucleotide in catalysis site and the presence of 3' to 5' proofreading or editing exonuclease activity. Thank you.